Hi, welcome to the third panel of the COVID-19 Symposium on strategic planning for crisis. The third panel is dedicated to vaccines for COVID-19, and obviously vaccines today are something that we have and we are deploying every day. But to go back probably 1 year ago, January, February 2020, when the SARS-CoV-2 virus started to basically be in China, get to Europe, other places, all people really started to ask when we can make a vaccine. And I was asked many times that question. Dr. Fauci was asked many times that question. And the answers of people like me that have been doing vaccines for many years was, well, usually to make a vaccine takes 10, 15, sometimes 20 years. This is a pandemic. We are going to go faster. We have new technologies, but minimum is going to take 18 years to have a vaccine. Maybe it's going to take 3 years. Things don't go well. So that's what what we were saying just a year ago, predicting when vaccines will be available. Well, reality bypassed every imagination that we had. Vaccines were ready in 10 months. And before the end of 2020, we started vaccinating. So how did that happen? Well, I think it happened for two reasons. One was new technologies. And the other one was an incredible investment from the public sector. About technologies, I wanted you to look at this first slide. Basically, I think what happened was the transition from what I call analogic vaccines to digital vaccines. So far, for under 50 years, we've been developing vaccines in a way that I call analogic vaccines, because to make vaccines, we needed to grow viruses, to grow bacteria, to grow parasites, grow them in fermenters, modify them, kill them, inject, or take portions of them, or modify them and inject. So in order to make vaccines, we really need to grow the pathogens. Well, that was I call analogic vaccines, because you need to have the material to make a vaccine. Well, this time we didn't need the virus. The virus was sequenced in China. Chinese put the sequence on January 20, 2020 in the internet. And that sequence basically was teleported across the internet, every place in the world, and hundreds of labs the same day were able to download the sequence and make a synthetic gene and use that synthetic gene to make a vaccine without ever seeing the virus. So that's the difference. I mean, we've been going from analogic to digital because we didn't need the live virus in order to make a vaccine. Next slide shows what people did with basically with the synthetic gene. Once the people had the synthetic gene, they made 3 vaccines. One very fast by using the synthetic gene to make a synthetic vaccine made by synthetic messenger RNA. The other type of vaccine, the synthetic gene, was put into a viral vector, typical adenovirus, and that was grown, that was used as a vaccine. And the third type of vaccine at the bottom of the slide, basically, the synthetic gene was used to engineer a cell, mammalian cell or insect cell or a tobacco cell, and that engineered cell will make a protein that will be purified, combined with an antigen, and used to make a vaccine. So these are the 3 types of vaccines. We'll learn more about them later in this panel. So that's why we went very fast. The next thing is why we went so fast. And the next slide shows what happened. basically, usually to make vaccines, we do things sequentially. First, we do discovery, then to phase 1, to phase 2, then phase 3. And in between, we pause, we look at the data, and that takes a long time. This time, and I mean, the pre-COVID vaccine development took between 10 and 20 years. This time, basically, everything was done in parallel. It was done in parallel because the governments, the public sector, the BARDA in particular, in the United States, spent more than 12 billion, which was given to companies basically so that they, to de-risk their investment, so they could do things in parallel. So discovery, phase 1, phase 2, and phase 3 were all done in parallel. And so things went extremely fast. And that's the reason we had the vaccine in 10 months. But beyond the technologies and the investment, we needed people. And the people that we have in this panel are the ones that basically actually made a lot of these things happen. It's with my pleasure that I'm going to introduce the people I'm going to talk in this panel. The first one is Barney Graham. Barney is at the Vaccine Research Center at the NIH, has been there since some time. And he has been working in many vaccines. He made a breakthrough in respiratory syncytial virus, and has been he basically designed the vaccines or the construct of the spike protein that's been used by 90% of the vaccines have been developed. So Barney, it is your turn to tell us how you designed the vaccine for COVID-19.
Thank you, Rino. And it's my pleasure to be here with you today. I'm going to just briefly review what I talked about in the recorded message that's online. First, I just want to review the history of vaccines in one slide. And Rino already mentioned much of this, but the first two vaccines were made for viruses were made by growing virus in animals, either on the hide of an animal for smallpox or in the spinal cord of an animal for rabies. When Goodpasture discovered how to grow virus in eggs, that allowed yellow fever and flu to come along, and the next cluster was because Enders discovered cell culture and allowed large amounts of virus to be grown up for either live attenuated or whole inactivated. Then there was this era of molecular biology in which you could make reassortants or virus-like particles or recombinant proteins. And as Rino said, this took us up to bioreactors. And recently we had a gene-based antigen delivered by a VSV that was an approved vaccine now for Ebola. Now I think we're going into a new era of structure-based vaccine design, and we thought that RSV might have been the first one for that, but COVID snuck in between. And COVID is based on some of the principles that we learned during the RSV vaccine development, but it also took advantage of all these other new technologies that have come up really over the last 10 to 20 years. And I'm just going to show how this has been built, and we won't stay here very long, but a lot of this has been built on technologies that have been used to try to make an HIV vaccine over these last 40 years. And even though we haven't made an HIV vaccine, this technology that has derived from that effort is largely what's behind the current advances. We worked on RSV for a long time on the enhanced illness syndrome and created an immunological paradigm that would allow us to get through the regulatory pathway. there was many advances in human monoclonal antibody discovery, and we've had what I consider inadequate or failed attempts at pandemic threats when chikungunya came through and MERS and then Ebola in 2014 and Zika in 2016. That started us thinking a lot about prototype pathogen approaches that would be more comprehensive. And we started looking for ways to combine structure-based design or precision vaccine antigen design with rapid platform manufacturing. So our work with Moderna started in 2017, looking for prototypic approaches to pair myxoviruses with Nipah and MERS coronaviruses for the coronavirus family. And when this outbreak started, we already had a way of stabilizing the spike protein, delivering it by mRNA, and protecting animals from lethal challenge. So we were in a position not only with the technology, but with a roadmap and with a pre-established private-public partnership that was effective. So this is what put us in the position to go fast. And just now looking into the future briefly, these are the things I think are the things that we really need to understand to break through on vaccines like influenza, HIV, herpes viruses, etc. So, and hepatitis C in particular. So antigenic variation and understanding how viruses can maintain persistence and have avoidance strategies to be able to persist in the host. And a lot of the antigenic variation creates a problem of immunodominance or immune avoidance that we have to learn how to induce subdominant lineages of antibodies. And it continues to into inducing memory cells that can maintain over longer periods of time and in the right places, meaning if it's a respiratory disease, to have tissue resident cells in the lung. And I think we need to continue to improve our approach to engineering and delivery of vaccines, both for stability and cost, so that we can move these technology into developing country settings. So I'll stop there and keep going.
Well, thank you, Barney. And really, thank you for this presentation, but thank you for really designing the vaccine for COVID-19. The next speaker is the one that basically is Larry Corey, is in Seattle, and it is the one basically has designed and coordinated all the major trials to make the vaccines available for people. Basically coordinating all the trials in the US, making sure that we have efficacy, making sure that trials were done quickly, making sure that trials were done with all the vaccines were becoming available. Larry has been conducting trials in the field of HIV for a very long time, as far as I remember, and basically has been using his expertise and the networks that were set up to do vaccine trials, basically to do a remarkable, incredible work to make these vaccines available to people in such a short period of time. Larry.
Well, thank you, Rino. Again, it's a pleasure to be here, and I'm going to just show a couple slides here first that sort of summarize sort of where we are in the vaccine field. I would say that, just in reflection, that I think what the clinical trials program has shown is that when the scientific community, both academia, industry, and government, can get together and really work together, one can essentially get unprecedented speed and efficiency. Obviously, the large public investment made a difference. It allowed us to work with companies on an individual basis, but yet set up a structure that we could harmonize the endpoints. We could have common clinical trials organizations. We have common DSMB, really the unsung heroes of the program, and set up a sort of a system that allowed each trial or each vaccine, each platform to be individually evaluated. But yet when we come out of it, we know that there's some reasonable commonalities that we can get a sense of, sure, does it work, and how well does it work? It is this working together that I think we need to carry through in the future with respect to the pace and the cadence. And if you have that kind of pace and cadence, when you have success, how it can implement things. I mean, when we designed the trials, we thought from beginning to end it would take 6 or 7 months to do this. It actually ended up being 4.5 months. Now we had an amazing epidemic also that doesn't necessarily be a great thing, but the reality is that we finished these trials a couple months earlier than we even planned, with remarkable success. And we essentially made the trials large. We almost doubled the size over what we needed to get better safety data, get more speed. And I think the trials have been incredibly effective with respect to predicting real-world efficacy. The high efficacy demonstrated in the large clinical RNA vaccine trials appeared to be holding in the population rollout. It was largely based on the Israeli data, which shows high efficacy even with the B.1.1.7 variant, which is very gratifying. There's a large 300,000 person, 500,000 person trial of the J&J vaccine that is ongoing now in South Africa and the healthcare workers, and that also is starting to show real-world efficacy similar to what we saw in the clinical trial. So the recent data suggests the vaccines are having an effect on hospitalization and mortality. The more recent data say that some of these vaccines appear to have good activity against variants of concern. I think there's an emerging set of data that highly potent vaccines, RNA vaccines, the J&J vaccines, probably, and what appear to be the protein vaccines, at least the Novavax vaccines, all, not only are better than natural infection, but they actually have some widespread efficacy against variants. And I think rather than thinking about herd immunity, I really think that we should be talking about vaccine coverage or vaccine-induced immunity, because vaccine coverage contributes the best approach to also not only protect against disease, but likely reduce variant spread. I think the real issue is obviously vaccines in bottles don't abrogate epidemics. Vaccination does. An extension of the best vaccines into the global population is what I think is our next need. Yes, it's a formidable task, but we have to create the infrastructure to do this. We need to recognize that giving vaccines that are only partially effective is really not going to be effective. I think the epidemics in Brazil and Chile and South Africa, in which we have neutralization-resistant variants predominate, are showing that high natural immunity and the use of less potent vaccines is really not an effective approach. And we must bring a way of having more effective vaccines to the epidemics. I think we've learned enough so that we need to narrow down the choices that we are making available when we use vaccines on a large population basis. Rino, your meningococcal vaccines, they were used in lower and middle income countries. We didn't look for, quote, less effective, potentially cheaper alternatives. We want to give the best vaccines to the best people. I think one of the things we have to recognize is that immunosuppressed patients who have chronic COVID are the major source of what we call multi-mutational variants of concern. And we need to actually direct vaccinations to immunosuppressed persons. And the largest population of immunosuppressed persons globally are HIV-infected people, especially those untreated or having ART breakthroughs. That's 24 million people living with HIV in sub-Saharan Africa. In South Africa alone, there's 7.5 people living with HIV. They have a heroic antiretroviral effort, but there's still 200,000 new infections yearly. And we need to develop effective vaccination regimens for these people. Whether that is J&J or RNA, or we need to think about making these vaccines more available to key countries where resistant variant epidemics are going to occur. Otherwise, we're going to have more resistance, and we're going to have the promulgation throughout the globe for this. And we need to think about creating manufacturing and supply chain and distribution of the highly potent vaccines globally. So I think that the neat thing is we've had a success story. We have effective vaccines. We know when we get good vaccine coverage, we can start making major advances on this epidemic with respect to hospitalization and death. But we certainly need to extend this globally, and that is going to require a concerted effort globally.
Well, thank you, Larry. Very compelling how vaccines were tested, how effective they are, but the big problem that still remains, which is basically how do we deploy these vaccines and produce these vaccines for the rest of the world. And as you said, designing vaccines by Barney is great. Showing that they're effective and safe is fantastic. But unless we inject those vaccines into people, they are not effective. So the third talk about Amanda Cohn from CDC is basically focused on taking the effective vaccines and making sure that we will be able to immunize people, which is an incredible task. Vaccines need to produce, need to be deployed. The organization needs to be incredible. The supply chain needs to be coordinated, and people need to be reached and injected. So, and everything has to be planned to be effective. And that's what Amanda has been, and her colleagues at CDC have been doing. So, Amanda, tell us how you did it.
Great. Thank you so much, Rino, for having me here today. And I'm going to wrap up these presentations with talking about not just the amazing work that got us these, right now, these 3 authorized vaccines in the United States, but actually the effort to get the country vaccinated. Excuse me. We have delivered, we've administered over 215 million vaccines in the United States since the middle of December to this week. So this is over a period of about 20 weeks, which is just really a remarkable success. We've achieved 80% coverage in adults age 65 years and older, and that's at least with a single dose. And over 65% of adults age 65 years and older are completely vaccinated. And if you look at just adults over the age of 18, more than 50% of adults in the United States have initiated vaccination. And so we are getting to, we're getting closer to vaccine immunity, but we need to get even further. This slide demonstrates the incredible amount of work and changes that have been done over the last 20 weeks, primarily at the jurisdictional and the local public health level. What you see here in gray is most vaccines have been administered in local health clinics in these vaccination sites that that local public health and state public health have set up. And then in yellow, what you can see are doses that have been delivered in pharmacies. And we have several federal programs where we've tried to augment the jurisdictional vaccination through places like federal retail pharmacy programs, and especially through HRSA, our federally qualified healthcare centers. Right now, we're also vaccinating in dialysis centers and in other places, really trying to augment in order to achieve vaccine equity, to make sure that no person is left behind and that we can really get vaccines into communities. I apologize. My dog has not barked this entire time until now. This slide shows how many vaccines we're giving per day. And what you can see is this has steadily increased over time. We've increased the number of channels that vaccines being delivered to. And up until last week, you could see we were steadily given, we were for the previous several weeks giving more than 3 million doses per week. Last week was the first week that we saw a decline in vaccination in terms of the total number of people who got vaccinated over the course of the week. And there is clearly the contributory factor of the pause in the J&J vaccine, which was called on by FDA and CDC last Tuesday. That pause was lifted yesterday, so we're excited about getting some of those doses that people had appointments for last week, getting vaccines in arms, either for J&J or for one of the mRNA vaccines this week and the upcoming week. But we also were seeing before that pause some leveling out of demand. And we really have to, this is a really critical time. I think the pause demonstrated that the United States is taking vaccine safety. Safety is the first priority. And we've delivered over 200 million doses of the mRNA vaccines with no safety signals detected. And when we quickly identified a safety signal with the J&J vaccine, FDA and CDC paused vaccination, and we spent 7 to 10 days making sure we understood everything we could about that safety signal, who was at risk, what was the degree of the risk, and looked at the risk-benefit comparison. And that's what was presented yesterday when the decision was made by the Advisory Committee for Immunization Practices to resume vaccination, making everyone aware that there is an increased risk for thrombosis with thrombocytopenia syndrome, which is a very rare thrombotic event that does appear to be associated with the adenovirus vector vaccine, J&J as well as the AstraZeneca, but is still a very is occurring rarely. So in women about 18 to 49 years of age, it's occurring at about 1 per 125,000 women getting vaccinated. It is really critical as we move into this next phase of vaccination that we continue to build trust both in communities and across the United States, that we bring vaccine as close as possible to people. We want vaccines to be in every community. We know that healthcare providers play a critical role. So as we have more doses of vaccine, we want to make sure that primary care providers are vaccinating and that people can get vaccine through their healthcare providers, which is really some states are relying on their healthcare providers quite a bit, but that needs to be more generalized throughout the United States. And we're really working closely with community partners, community-based organizations, places like faith-based organizations, churches, local rec centers to sponsor vaccination clinics, either with pharmacies or with local health systems, to really make this a community effort to get the country vaccinated, community by community. And, we've made amazing progress. We're protecting older adults who are at most risk for hospitalizations and deaths. And we've seen hospitalizations and deaths decline in this older age group, but really to have the type of total impact to benefit from these vaccines, both the direct and indirect protection that happens when you achieve high coverage, we need to get the entire country vaccinated. And the next big step in that will be in the coming weeks or months when we anticipate vaccines being available for younger children, going down first probably to age 12, and then hopefully over the coming months available for all children. And I'll close there. Thank you very much.
Well, thank you, Amanda. That's impressive what you have achieved and what you've been done. I think we got at the end of our 3 presentations from our 3 panelists, and now we will start the discussion. I'll probably start with the first question, which could be for probably Larry and maybe Amanda as well could talk about it. And because we heard an incredible story. The incredible story is that vaccines have been done well, they work well, they've been developed, shown they're efficacious, and deployed, and with the impressive figure of 250 million doses delivered in the United States. Incredible. So we have a beautiful, incredible story. At the same time, we know that this world, the world is basically 8 billion people. And Larry said, I mean, we need to vaccinate all of them. But we are not going to do that quickly. Some people say we don't have enough vaccines, and people say it's going to take up to 2023 before we can vaccinate all people globally. So the question is, we've done something which is remarkable, but we could do better if we. What will it take to do better? To be ready to make people globally faster.
Well, I guess I have to say that, this is where government and industry and the world need to get together. I sort of look at the issue of what we had with HIV and antiretroviral therapy as a model that we need to move to the model of sort of the concept of transferring the technology, tiered pricing, the development of the sort of distributive, sort of generic production of the high potency vaccines. So we get them distributed and made, not only for this epidemic, for the next epidemic. The ones that using the platforms that have staying power and that could actually be used over time to sort of force the investment. So I do think the concept of taking lower income countries and vaccinating only 20% of the persons is a flawed concept. And I also think the concept that we should have 30 vaccines or 40 vaccines is also a flawed concept. I think that we need to concentrate our resources on highly active vaccines and figure out ways to get them distributed, manufactured and distributed as best we can. It's in our interest. I often say that, a generation is defined as Barney and you have said, by not only by inventing its technologies, but how it uses its technologies. Our humanity is defined by how we use our technologies. And I think this is really one epidemic that's causing death in everywhere. It has no borders, and it is really, and the economic benefit has been unbelievable. It's the pandemic of the last 100 years, maybe the pandemic almost forever, and it's absolute cost. And I think we need to bring resources to bear to globally to put this under control.
Yeah, I guess in the HIV that you mentioned was the PEPFAR, basically, project, where basically it was a huge investment from the United States that allowed the transfer of the technology to low-income countries that manufacture their own drugs. This is a scientific panel, but should we suggest something similar to the world?
I would. I say the road has been, we have a path. We created a path. Let's reduce
and has been successful. Yes.
Right. We have Gavi. We have global funds. We have the ability to do that. We need to build the production model. And yes, that will take some time. But we also have models in which we can increase production of our currently most potent vaccines and get it into people's arms as much as we can. And the variants, the virus is telling us that we need to do this. Barney might reflect on that, but we're looking at these countries that have an enormous number of seropositives who are getting reinfected with still severe disease. So we have to hit them with vaccines that actually do work and prevent severe disease.
Absolutely. Well, thank you, Larry. Amanda, I don't know if you want to say something about this. Otherwise, I have a question, obviously, for you. But otherwise, I'm going to go to Barney. Barney, we've been talking about variants, about vaccines that are they seem to work. Well, we know the present vaccine, some of them don't work or work much less against the variants. But some vaccines seem to work, but obviously second generation vaccines are already in the clinic. So hopefully we'll be able to control the variants which are out there. What's your feeling? We're going to have universal vaccines, at least for COVID-19. Is that possible? Or we'll have to basically every few years to adjust the vaccines again?
Yes, there's several parts to that question. I think as the epidemic or pandemic progresses over time, I'm hoping that it will become a seasonal disease. And then eventually it'll become a childhood disease, just like our other endemic coronaviruses. And so I think the types of vaccines, the need for vaccines is going to change over the next 3 to 20 years. The variants, I'm hoping, I don't know this, but I'm hoping that a lot of the initial change has been for transmission's sake and not for immune escape. And all the changes that have happened so far, even in the most difficult variants, like the one from South Africa or the one from India now with the 484 and 452 changes, I think that each of those amino acids can be tied to an improved infection or transmission, phenotype. So I'm hoping that this levels off and it's not like influenza, because for influenza, that's where you get into the antigenic variation, the immunodominance problems, some of the real difficult immunology that we really haven't solved yet. There are technologies, though, and several groups are now approaching this using nanoparticle display. You can make polyvalent or multivalent nanoparticles and cover a lot of different coronaviruses. I think it's going to be relatively easy to make beta coronavirus vaccines. Whether we can get all the alpha viruses and other coronaviruses, Delta virus hasn't infected us yet, but it might. And so I don't know if we'll get them all in one particle, but I think it's feasible now to make a pan coronavirus vaccine. But I think the real question goes back to what Larry was saying in your first question. We have to get the vaccines deployed so we can stop transmission, so we can stop the variants from emerging. If we can stop transmission, the variants will stop emerging. And so to me, that is the key step we have to take. And I loved your slide, your analog to digital slide, because this is the way I envision it. You skip a couple of generations of technologies for manufacturing and go straight to chemical synthesis and get this distributed around the world, just like we did for cell phones.
Well, thank you. And it's actually encouraging that you say we're going to have a universal vaccine. Actually, I believe it's possible too. just need to continue working and watch. But as you said, I mean, if you want to stop this virus and stop the variants, we need to vaccinate people globally.
Yes.
Well, thank you. I think now we're going to go to some of the questions I had in mind for Amanda, but it's also coming from the audience. And obviously it's about safety and the clotting problem, the thrombolytic events. Amanda, you mentioned that already about the J&J, what the ACIP recommended yesterday, but there are still a lot of questions out there in the mind of people. And one specific question which is out there for the audience is whether the clotting problem is the same for J&J and AstraZeneca. And probably you want to comment a little bit broader about this.
Sure. And just before we go to that, I do want to say, we have to get vaccines deployed globally, but we also have to work with business, communities, and government to stop misinformation, because that is going to be, we can't get people vaccinated, even if they're available, if we don't figure out how to make sure that people really understand these vaccines and have the information they need to understand the risks and benefits. So the J&J, the thrombosis with thrombocytopenia, this signal that was detected in the J&J vaccine, in part, we were able to detect it so quickly. These cases are incredibly rare, so our vaccine safety signals would have detected it quickly, but we were super aware of it because there had been hundreds, or I don't remember the exact number, but dozens and now hundreds, I believe, of cases similar to this with the AstraZeneca vaccine, which is also an adenovirus vector vaccine, but they're different adenoviruses. So we don't totally understand why both of these vaccines are causing what is essentially being seen as the same syndrome. But it is likely, it is clearly very, the prevalence is incredibly low. It seems to be an immunologic autoimmune type of event. And these cases, if they're treated with IVIG, that is the treatment that these patients are recovering with. So essentially, the syndrome is being looked at similarly. We don't have the AstraZeneca vaccine in this country, so we haven't looked at the data from that vaccine as intensively at ACIP or at CDC in the same way that they have in Europe. But all of the results, and including the lab findings that are identifying the platelet factor 4 antibody and others are similar in both vaccines. I don't know if anybody else has anything more they want to add.
Well, I mean, the clinical syndrome, I think, is the same. These really very significant thrombotic events, often in the brain and the cavernous sinuses, but also elsewhere, associated with a high platelet factor 4, sort of a syndrome that's sort of like heparin-induced thrombotic thrombocytopenia, but of course heparin is not to be used. That actually makes the person's worse. So I think there are differences. There are more males associated with the AZ vaccine. I think it's been very hard to get numerator and denominator. There's been a lot more AZ vaccine, and there are well over 200 cases now in Europe. But the incidence, to be really accurate as to what is one better than the other is really unclear at the moment. And I think we just need to get down to understanding that better.
Thank you. We've got 5 minutes left, and we got 3 more from the audience. They're pretty simple questions, so please try to answer quickly so that we can do all of them. The first one is for Barney. And how does a single dose immunization with RNA vaccine, Moderna or Pfizer, compare to the J&J vaccine? Barney, are you?
Yes, it depends. It depends on if you're measuring antibody responses or T cell responses or clinical efficacy. There is single dose efficacy with the RNA vaccines. You can see a separation of the curves before the second dose, but it's clear that the second dose does bring almost everyone up to the same level of antibody, and so it makes it a much more uniform immunity across more people, and it's likely to be more durable. And so I think the second dose does help the uniformity, the magnitude, and the durability. But there is protection after a single dose of RNA. We don't have a way of comparing it for clinical outcome because they were tested as a 2-dose vaccine. We'll know more when J&J finishes their testing of the 2-dose J&J vaccine.
Thank you, Barney. Well, the second question is, can we discuss what's the most effective regimen to protect against variants, booster doses, new vaccines, whether the vaccine induced lower antibody titers in the elderly? Larry, probably that's for you.
I mean, I think real world effectiveness is just starting to come out. In the really elderly, we don't have a huge experience. We didn't enroll that many people greater than 85 into the clinical trials. So we do know age has a factor here. So I think we just have to wait and see as to what will happen. I think booster doses are being looked at. The memory responses at 6 months look good. The data from Pfizer and some preliminary data that's not yet published from Moderna all suggest that the high efficacy in the 90s is holding for up to 6 months, and the antibody responses are holding up to 6 months. We know that there have been studies to show that there's a reasonable amount of B cells in memory. So there are things that of this wheat field that we have not cut down yet. We're learning as we go. We have very large cohorts now that will teach us as we go, and we have to set up monitoring systems to see if failure occurs, and if that a failure occurs is because of waning immunity or variants. I think from my point of view, the Israeli data has shown 1 dose with Pfizer is like 40% effective. 2 doses in the real world is like 90% effective. Well, I want the 90% effective. I'm worried that the 1-dose vaccines in an area where there's variants are going to actually speed the emergence of escape-resistant variants. And I think that the regimens have to be what the clinical trials were based on, which is 2 doses for the RNA and going to be 2 doses for the protein, and we'll see how the rest of them work out.
Thank you, Larry. We have one last question, which I think probably is also for Amanda, which is basically, what is known about the long-term effects of vaccines? How do you convince people who are worried the possibility of serious long-term consequences? How can we convince them to be vaccinated?
So just to build off of what Larry was saying, we have many studies in progress to evaluate how long these vaccines will last for. And we have good data already showing that these vaccines there's good memory response at 6 months, but real world effectiveness, we have 4 to 5 months of data, and we are definitely seeing breakthrough cases, but we would anticipate seeing breakthrough cases. Even if a vaccine is 90% effective, when you have so much transmission, you're going to see cases in people who have been vaccinated. In terms of long-term safety, what I will say, and this is what I say to so many people, is we know that COVID causes, in many, many people, I think it's 16% of people right now, long-term symptoms and problems, neurologic, pulmonary, heart problems, and those symptoms can have serious impact on people's lives, and we've seen that. We have never really, there's no examples of vaccines ever causing problems that haven't been, safety signals that haven't been identified 6 to 8 weeks after vaccination. We vaccinated 200 million people with these mRNA vaccines, and those have included people from all walks of life. It's included pregnant women, it's included older adults, younger adults, and many of those individuals were vaccinated 4 to 5 months ago, and we've detected no signals. And we're following people. We have VSafe, which is an after vaccination checker, which continues to text people monthly to see if they've had any signs or symptoms of anything. And so I think the plausibility or the likelihood of there being a long-term adverse event associated with these vaccines is incredibly low. Nothing is, there's always some risk, but it's incredibly low. And we know that for people who do survive COVID, that there are long-term consequences in a high proportion of those people.
Well, thank you. I think we reached the end of the time allocated for this panel. I really like to thank Barney, Larry, and Amanda. I think you've done a fantastic job, and it's really a privilege to have in a panel the 3 people that really made all these things happen. I really want to thank you. Also want to thank the audience and the National Academy of Science allowed us to have this conversation. I think this is the end of this panel. We're going to go on with the 4th panel in 10 minutes. Thank you very much to everybody.
Thank you.
Pleasure.
And thank you,
Thank you, Rino.
Thanks for the nice moderation, Rino. Thank you.